In this video, we are going to cover the topic cardiovascular system. Two topics we are going to look through: the general characteristics of cardiovascular system and properties of cardiovascular system. Before diving into this topic, let us first briefly describe the structure of heart, the basic part. So, heart is made up of four chambers: right atrium, right ventricle, left atrium, and left ventricle. And this atria is separated by interarterial septum, and this ventricle is separated by interventricular septum. So what we read just now, heart is made up of four chambers: right atrium, right ventricle, left atrium, left ventricle. So atria is separated by interarterial septum and ventricle is separated by interventricular septum. Okay, now this is done. Now main thing to remember here is ventricle are always thicker than atria. In every case, like in right ventricle also, in left ventricle also, ventricles are a little bit thicker, more thicker than the atria, because force of contraction of heart depends upon the muscle. So after this, let's move on to the layers of wall of heart. So heart is made up of three layers: outer pericardium, middle myocardium, and inner endocardium. This outer pericardium is also divided into two layers, that is outer parietal pericardium, inner visceral pericardium. We are not going deep into this. Just to remember what is the main purpose of the outer layer of every structure generally is to protect. So here also the outer layer of parietal pericardium is to protect or protect the heart basically. Inner visceral pericardium is also has two layers. It's made up of two layers: outer fibrous layer, inner serous layer. Outer parietal pericardium. Inner serous layer, our main function is to provide serous fluid, which provides friction between both the layers. And total volume of this fluid is 25 to 35 ml. Moving to the next layer, that is middle myocardium. We all know heart is made up of cardiac muscle, okay, and cardiac muscle and skeletal muscle both are same. So how can we differentiate between skeletal and cardiac muscle? The best way to differentiate is that skeletal muscle is not branched, and cardiac muscle is branched. So cardiac muscle is branched and skeletal muscle is not branched. Now all this we have already studied in previous physiology topics about this actin, myosin, troponin, tropomyosin, all these proteins and all, sarcomere, myofibrils and all. So nothing to repeat. The one important term that is also given in the PPT is syncytium. It means tissue with cytoplasmic continuity between adjacent cells. Now why it is important? Heart is physiological syncytium. Why is it so? Because it has no continuity of cytoplasm, and muscle fibers are separated from each other by cell membrane. Okay, and the membranes of adjacent muscle fibers fuse together to form gap junctions. Okay, and why this gap junction is important? It is permeable to ions and facilitate rapid conduction of action potential. This term is, you already might have read about action potential and all. Now in myocardium, 3 points you have to remember. First of all, 3 functions myocardium has. First of all, contractile function. How does it conduct it? Like through the proteins in muscles. Okay. Second is the pacemaker. Heart is the pacemaker. Now as soon as what is pacemaker basically? That it generates impulse for heartbeat, and it is performed by P cells. What is P cells? Pacemaker cells. And as soon as I say the word pacemaker cells, you should automatically come to your mind the sinoarterial node, which is the main pacemaker in human heart. So pacemaker SA node is conducted by SA node. And third function is conductive system. Okay, it is formed by cardiac muscle fiber. Impulse from SA node are transmitted to atria directly. Okay, detail will be explained in next chapter. Now let's move on to the last layer, endocardium. Endocardium, the main topics to cover is valves. Now, there are two types of valves: arterioventricular valves and semilunar valves. Arterioventricular valves have two valves, that is bicuspid, also known as mitral valve, and tricuspid valve. Now, these valves are attached to papillary muscles by means of caudal tendinae. Now, where is papillary muscles here? This is papillary muscles. It is attached by caudal tendinae, or whatever, however it's pronounced. Semilunar valves, we have two valves: aortic valve and pulmonary valve. Now, where it is found? Okay, let me first just see this image. This is right atrium. What is this? Number 4 is tricuspid valve, and here we can see the mitral valve or the bicuspid valve. Here will be the pulmonary valve, and this is the arterial valve. Okay. Now, let's move on to the circulation of heart. So, blood enters the right atrium by inferior vena cava and superior vena cava, and it enters the right ventricle. From there, blood is pumped through, and then it is branched into two parts. What is this? Pulmonary artery is branched into two parts, and it goes to the right and left lung. From there, the deoxygenated blood becomes oxygenated and then comes into left atrium. From there, through, what is this valve's name? Mitral valve or bicuspid valve. And then it goes through the left atrium. From left atrium, the aorta supplies to upper and lower body parts. This is a graphical representation of how blood is pumped. So you can see here, the deoxygenated blood comes through the inferior and superior vena cava. The blue color arrow goes to right atrium, and then pulmonary valve goes to the lungs, comes as oxygenated blood in the left atrium, left ventricle, and then goes through aorta and supplies the whole body part. What is the function of cardiovascular system? There are two main functions: hemodynamic and non-hemodynamic. So hemodynamic means provides movement of blood along vessels, and non-hemodynamic means endocrine function, basically to control
Homeostasis means
control of temperature. Moving to the next topic, that is the properties. There are 5 properties: excitability, refractivity, conductivity, contractility, and automaticity. So let us cover one by one. What is excitability? First of all, let's discuss the definition, that is ability of a living tissue to give response to a stimulus. If a stimulus is generated, then some response should be there, right? So that is known as excitability. The name itself tells to excite something. So here this value is important to learn. Resting membrane potential's value is minus 90 millivolt. Amplitude is 120 MV. Duration is 300 millisecond. Now there are two phases of electric potential. Now so action potential. It can be through electrical or ionic. We are going to discuss about both the topics. First of all, let us see the electrical potential. This is cardiac action potential. So first we are in a rest phase. And then once it crosses the threshold, there is a sudden depolarization. The depolarization is there. Now what is the reason? The reason is hidden in the ionic part. So first let's just see the electrical point of view. So in this depolarization, it's very rapid and it just takes 2 milliseconds. So you can imagine how fast it is. Now the next is after depolarization, it's fast initial repolarization. It is also very rapid. It also takes about 2 milliseconds. And the end of it is represented by a notch. The second is slow repolarization. So you can see the duration. It's very slow. So you have to see what is slow, what is fast and all. You have to remember. This is slow repolarization, or we also call it as plateau. The muscle remains in depolarized state for some time before further repolarization. So it forms a plateau, that is a stable period in action potential curves, and it lasts for about 200 milliseconds. And time is also longer in cardiac muscle, by 5 to 15 times than in skeletal muscle. Number 3, that is this graph, is fast final repolarization. You can see the time, how quickly it's descending. So this is final repolarization.
It occurs after the plateau and it is kind of a fast process compared to the last one. Takes about 50 to 80 millisecond. Then comes the re-establishment of resting membrane potential, rest phase.
So this was the electrical potential part. Moving to the ion efflux of potassium, calcium, etc. We are going to see one by one. So first of all is fast depolarization we studied. How does this fast rate depolarization occurs? It is because of the Na plus ions. Na plus ion channel will get open. As soon as it gets open, Na plus ions gets in. Okay, and this is fast influx of sodium ion. So since sodium ion gets in so quickly, that's why it's fast depolarization here. Next is initial repolarization. It is due to the potassium channels. Potassium channels. Now it is the efflux, that is from this channels potassium ions will get out from the muscle fiber. Okay, the fast sodium channel close suddenly and slow sodium channels open, resulting in slow influx of low quantity of sodium ion. Okay, so slowly Na plus channels gets closes and the K plus ions opens. So it's like slow process and little bit slow process. Next, moving on to the calcium channels. Calcium channels is very slow. These channels are kept open for a long period of time and cause influx of large number of calcium ion. So already the slow sodium channels are open, through which slow influx of sodium ion continues. Because of the entry of this calcium and sodium ions into the muscle fiber, positivity is maintained inside the muscle fiber, producing a prolonged depolarization. Okay, that's why the time of depolarization is 200 millisecond. Now moving on to the third phase, that is the repolarization phase. From here what happens? Efflux of potassium ions. Number of potassium ions moving out of the muscle fiber exceeds the number of calcium ion moving in. So now negativity is inside and positivity is outside, resulting in final repolarization. And then comes the resting phase or the resting membrane potential. So here activation of sodium potassium pump is maintained. Excess of calcium ion which had entered the muscle fiber also move out through sodium calcium pump, thus resulting in resting membrane potential, and it is restored. Moving to the next, that is changes of excitability, is divided into three categories: absolute resting potential, relative resting potential, supernormal excitability or exaltation. Absolute resting potential is about 0.27 second. Relative resting potential is about 0.03 second. Just to remember the time, that's it. Now the refractivity, coming to the next property, that is refractivity. First of all, let us understand the term refractivity. That means muscle does not show any response to stimulus. And you have already read about twitch, muscle twitch, single twitch, complete twitch, incomplete twitch and all. So this refractivity is basically what, when a muscle does not show any response to a stimulus. Yeah, and it prevents tetanic contraction. Now, first of all, what is a single twitch? What is multiple twitch? We will understand. Like if you give one impulse, then it will be like single twitch. But if you give like continuous impulse, it will be like, okay, this is what complete tetanus. Incomplete tetanus is like if you give one single impulse, it will be like this. And if you give impulse here only, it will be very irregular, like this. This is incomplete tetanus. So coming back to the refractivity, important here to understand it, it prevents tetanic contraction, and muscle is a single twitch. See, this is the graph we need to see. This is a stimulus during systole, and here we can see it's literally no response, like zero response. And stimulus during diastole, we can see a response, but here it's also again we can see one impulse. Now this is what is known as extrasystole. Extrasystole or premature contraction means extraordinary contraction of heart after extraordinary excitation at diastolic period. After this comes the compensatory pause. That is, this extrasystole, first of all let's see, this extrasystole is of two types: arterial and ventricular. Now this arterial is nothing to see, it's not important. Ventricular here comes the important term compensatory pause. So after extrasystole, the compensatory pause occurs because next regular impulse come during absolute refractivity period. Continued is extrasystole. In short, it's just temporary stoppage of heart before it starts contracting. That's it to see. So if you are, it's difficult for you to remember this definition, just remember this one. Next, coming to the property conductivity. These 3 points you need to learn. What is conductivity? Basically, it's the generation of action potential, right? So the first main function of conductivity is to generate action potential, and conduction of action potential is second. Once it's generated, it will be conducted. That is known as propagation. Provide excessive contraction of heart. So because of this generation of action potential, contraction of heart of muscles will take place. And then this diagram is also important. We have to see where are the nodes, where are fibers and all. So this number 1 node which you see in right atrium is sinoarterial node. We have already studied in, like just now we have studied in that pacemaker cells make this SA node is responsible for conduction of impulse. It's very important point. Second, going to the AV node, that is atrium ventricular node. From there, this is the electrical part of the heart. So how electrically it is stimulated. Number 3 is bundle of His. Number 4, here you can see this is right and left bundle branches. And number 5 is Purkinje fibers. So electrically how it's conducted? From SA node, it spreads to like every part of the heart. From there, AV node collects, and then right and left branches. It goes from here to here through Purkinje fibers. Conduction velocity of action potential. Both atrium and ventricular muscles is 0.8 to
1 meter per second.
And bundle of His is 2 to 3. Purkinje fiber is 4 to 5. AV node, that is AV nodal delay, is 0.02 to 0.05. This is important here, AV nodal delay, because as you can see, it's very, very slow. Coming to the next property, that is contractility. First, the definition: ability of tissue to shorten in length after receiving a particular stimulus. So various factors affect the contractile properties of cardiac muscle. One important definition here is all or none law. So what is all or none law? Whatever may be the strength, the whole cardiac muscle gives maximum response, or it does not give any response at all. Below the threshold level, if the strength of muscles stimulus is not adequate, the muscle does not give response. So it was given in the first PPT of physiology about all or none law, and it is demonstrated by the heart of frog. By applying the first stimulus, you can see sinus, venous and right auricle. Ventricle is stimulated by placing the electrode at the base of ventricle. So it's just an experiment that one stimulus is applied with minimum strength of 1 volt at the base of ventricle, and contraction is recorded. Then after 20 seconds, the strength of stimulus is increased to 2 volt and stimulus is again applied. So the curve is recorded. The procedure is repeated by strengthening the volt every time, as you can see. So amplitude of all contraction remains the same. As you can see, there's literally no change. So what does this experiment tell us about? What is the cause of this experiment? It's basically the
Syncytial arrangement of
cardiac muscle. It's because of this arrangement that this graph is maintained, and all or none law is applicable. We come to this conclusion. The next and the last property is automaticity. It is ability of tissue to produce its own impulse regularly. So here we need to learn the impulse, like values here. First pacemaker, we all know, is the SA node. So its number is 60 to 80 impulse per minute. Second pacemaker is AV node, that is 40 to 50 per minute. Bundle of His is 30 to 40 per minute, and Purkinje fiber is 20 per minute. Now there are certain theories of automaticity. After this we will be done with the presentation or the video. Okay, so theories of automaticity is 2 types. First is neurogenic, second is myogenic. We all know that conduction of blood is because of electrical impulse also, and because of due to muscles also. So these are theories of automaticity. Coming to the pacemaker potential or slow diastolic depolarization. The polarization of membrane changes during this phase. When it achieves threshold potential, the action potential occurs. So this is the pacemaker potential here, and this is the action potential here. So what is pacemaker potential? Is the unstable resting membrane potential in SA node. So this is the unstable resting membrane potential in SA node. It is also called prepotential. Okay, electrical potential in SA node is different from that of other cardiac muscle fiber. Remember this. In SA node, each impulse triggers the next impulse. It is mainly due to unstable resting membrane potential. Now, what is the cause of this slow diastolic depolarization? Initial part, we all know this graph is due to the ionic mechanism occurring here. So let us just see the reason behind this. So initial part is due to the slow influx of Na plus ions. Okay, because of the Na plus ion channel opens slowly and then Na plus moves in very slowly. Because of this, the part is slower here and it takes a lot of time. And the later part is due to the influx of calcium channels. So again, to recap everything, resting membrane potential is not stable in the SA node. Okay, to start with, the sodium ions leak into the pacemaker fibers and cause slow depolarization. Okay, this slow depolarization forms the initial part of pacemaker potential. Then comes the calcium channels role. Okay, it starts opening. At the beginning, there is a slow influx of calcium ions causing further depolarization in same slower rate. It follows the later part of the pacemaker potential. Hence how this graph is formed. Okay, pacemaker potential, action potential. Action potential, pacemaker potential. Depolarization, repolarization. Okay. So the initial part of pacemaker potential is due to slow influx of sodium ions and the later part is due to the slow influx of calcium ions. Done. So this is all about the cardiovascular system. Two topics we have studied: general characteristics of cardiovascular system and the properties of cardiovascular system. Thank you.